All right, here we are. So normally we would do this in the second week of a semester as well. This is kind of the same speed for lab, just a little bit different speed for lecture. This would be my normal week 2. Of course, as you are reviewing this material, one of the things I always wanted to say is if you go to the very end of the PowerPoint, you're going to find two things here. Number one is a set of blank diagrams that will be used for exams as well. And then back here is another set of blank diagrams that I give. These also will be used on the examinations. So let's pick up with where we left off. We were just we had done the ear. Now we're on the eye. So in the eye, we're going to talk about the major things are the eye, the optic nerve, the brain, and accessory structures. Now today we're not going to be talking about the brain. We do in lecture when we talk about the occipital lobe being the primary visual cortex and how the lateral geniculate nucleus was the part of the thalamus that sends any of this visual information to the correct place after it comes from the optic chiasm and the optic nerve and the eyeball itself. And the eyeball, we talk about the things that do the visual stuff. Now, what we want to start off with, I'm going to briefly mention this. We did this in my lab as well. You'll note, or my lecture, what I a lot of times like to do is use my kind of similar things to my lab. So I cover that anatomy twice. Now, the eyebrows and eyelashes we talked about in lecture, we just said their eyes for their hairs for protections. We're not going to actually be able to see them, but an eyebrow and an eyelash should be a type of accessory structure. The palpebrae, that's your eyelids. There's an upper and lower, superior and inferior palpebrae. We'll see that in a moment. The canthus, that's where your palpebrae meet. There's a medial and lateral canthus where the superior and inferior palpebrae meet. Inside the inner lining of the palpebrae, there are the meibomian glands or tarsal glands. They are specialized sebaceous glands. They make lipids to keep the palpebrae from sticking together. And when they get infected, the ducts become infected and trapped. You end up getting a sty. We saw the lacrimal carbuncle in lecture. That's what makes lipids while you're asleep. It just keeps your so if you go to the medial side of your eye orbit, you'll find crusty material there when you wake up. Your sleep, your eye boogers, that's your secretions of lacrimal carbuncle in the medial canthus. Now we also know there's the two conjunctiva. There is palpebral conjunctiva. That's what's on the inside of the eyelid. And then it folds over on itself and forms the ocular conjunctiva. So let's take a look at that. Palpebrae. There is an upper palpebrae, upper eyelid, and a lower eyelid. The superior palpebrae and the inferior palpebrae. There are the canthus on each side. There's a medial canthus and a lateral canthus. Here they have them as angle in this diagram, but the canthus is the place where these meet. Okay. The tarsal glands are not depictable here. We don't have an image of tarsal glands. They are in there. They cover the inside. You can actually, a lot of times, some of the older images, the image I have in my book, tried to show the ducts opening up there and stuff like that. So, and anyway, as well, our lacrimal carbuncle. Lacrimal carbuncle right here, you can see very well here. And then lastly, the conjunctiva. Now to see the conjunctiva, here we have the bulbar conjunctiva and the palpebral conjunctiva. Palpebral conjunctiva, bulbar conjunctiva, or ocular conjunctiva. Okay. So that should get all of those things complete. Now, then what we want to see, and here's some, then what we want to see is the extrinsic eye muscles. Those muscles on the outside of the eye. They control the eyeball's position. They move the eye. As they're controlled by cranial nerves, cranial nerve injuries in the head can cause ptosis or drooping of the eyeball where it drifts, things like that. Weaken muscles, certain types of injuries. Also, one of the things you guys will be looking for is because the cranial nerves, that their nuclei are in the midbrain. Common thing is to check eyeball movement when you track it with a light. Like they take a light or their finger and have you watch it, and they're watching for those movements. They're referred to as saccades, and you'll be looking at that too. So, there's just a few important clinical reasons, part of neurological examinations, that you'll be learning about that you need to know these about. So, we'll see the superior rectus, inferior rectus, superior oblique, inferior oblique, lateral and medial rectus muscles. Now, these muscles control the position of the eyeball. If we were to look at the lateral side, the lateral rectus muscle runs along the side, which means the medial rectus is over here. Now, so what we've done is flipped it, and we're looking at the medial side. It's on the opposite side here. So you'd see medial rectus, lateral rectus. Superior rectus going straight back, and then the inferior rectus going straight back. So rectus means they go straight back. Then the inferior oblique travels sideways, and the superior oblique travels sideways. It goes obliquely or in an angle. Okay. So as we said, since it has that pulley shape to it, it's the only one that does that. That's the one that's actually controlled by the oblique, by the trochlear nerve, superior oblique. All right. Now, the lacrimal apparatus. This is the portion of the eyeball itself that is an accessory structure that goes through that helps produce tears, conduct tears, and remove tears to the nasal cavity. The tears are produced in a gland that is at the upper part of the eye, superior and lateral to the eye, referred to as a lacrimal gland. Lacrimal gland produces the tears. The lacrimal ducts take the tears that the lacrimal gland produces and takes them to the eyeball surface where it can wash the eye out. And then at the bottom of the eye, basically the space between the inferior palpebrae and the eyeball, we can call that the lacrimal lake. To drain that lacrimal lake is the pores called the lacrimal puncta. Two lacrimal puncta are attached to two lacrimal canaliculi. Canaliculi, canaliculi. The two canals, the superior canaliculi, inferior canaliculi, take the tears from the puncta and they go to the lacrimal sac. And the lacrimal sac then is the soft tissue that takes the tears into nasolacrimal duct, and nasolacrimal duct takes tears to the nasal cavity. And why you have to blow your nose when you cry. So if we look at this is the diagram I will use for the test, is our lacrimal gland up here, then draining into many lacrimal ducts that will go to the surface of the eyeball, washing out, like if you get something in the eye. Then it will go down here to the two lacrimal puncta, or punctum, the two lacrimal puncta, and then those enter to the superior and inferior lacrimal canaliculi, which go to the lacrimal sac, down to the nasolacrimal duct, to the nasal cavity. Okay, and this goes to your nasal cavity. Okay, so these are the major things there. And this is the diagram I'll use for the lacrimal gland, lacrimal ducts, all those things I just showed here. Now, there are other things in this image being displayed, but I just want you to make sure you know that's only what I'm looking at here. That's what I'm focusing on here, okay? And that's what I'm going to use that diagram for. Now, the eyeball, we've mentioned in lecture, if you watch the lecture first, we're watching this first, really depends on which one you prefer. The eyeball has three tunics. There's an outer fibrous tunic, a middle vascular tunic, and an inner more retina or neural tunic. The outermost fibrous tunic, middle vascular, innermost retina, neural tunic. And so one of the things you have is the fibrous tunic The vascular tunic is sometimes called the uvea, and the neural tunic is sometimes called the retina. Now, what happens is these divisions or tunics, as they divide up, have different functions. So let's look at the fibrous tunic first. And the fibrous tunic we know is outermost. Now, it contains two major structures. The sclera, the white part, and the cornea, the clear, transparent part. Now, I always like to say that eye puns don't get any cornea. The cornea, being clear, is a place that refraction happens, we talked about in lecture. Now, there is a corneal junction or corneal limbus between the sclera and cornea, and we'll see those labeled here, sometimes called the cornea-scleral junction. There is a scleral venous sinus or canal of Schlemm. Will the real Schlemm Shady please stand up? Delivers your aqueous humor that we produce by your ciliary bodies back to the veins to reduce interocular pressure and maintain proper pressure. Okay, so as we look at that, let's see our sclera, the white part of the eye, and the cornea, the clear part of the eye. Cornea, sclera. Now, the corneal junction or corneoscleral junction is where the cornea meets the sclera. It is the limbus, the junction between the two. Now, the scleral venous sinus. Here you can actually see that whole right there's scleral venous sinus. But as we zoom in, scleral venous sinus or canal Schlemm, right here we have ciliary body. These red arrows are showing the production of aqueous humor. Aqueous humor will go into this eyeball and be picked up by scleral venous sinus. When this is not working properly, that is what causes glaucoma. Okay. Now, that gets all those materials there. Now, the eyeball, the vascular tunic. Vascular tunic, we have an iris. That's the pigmented part. It means it's the colored part of the eye. We're going to see that it changes shape of the pupil. We're going to see that's what actually makes the pupil get bigger or smaller. The choroid, which is highly vascular. When you ever hear the word chorion, choroid, something chory, is talking about blood vessels, very highly vascular area. So the choroid is the extremely vascular portion. Then associated to the vascular portion of the vascular tunic is what choroid puts the vascular and vascular tunic, is the blood vessels and lymphatic vessels found there. You're going to see the pupil, the lens, and the ciliary body. Now, the pupil, the lens, ciliary body, they are a little bit different. One of the things we'll see is the pupil is the opening in your iris, and that's what light can pass through. Light can go and pass through that, and that light can then pass through to the lens. And the lens focuses it. Now, what does it mean to focus? Focus means I'm bending light so that it is going to be a clear image. So focusing on the photoreceptors of your retina. Remember, photoreceptors are the specialized neurons who turn light energy into nerve impulses. So basically, think of photoreceptors of the retina. The retina is somewhat like an old film that you exposed in a camera. Think of a photoreceptor as each pixel of your digital cameras in your phone, that every pixel is one photoreceptor, and the light that hit that pixel is that color. And you put all the photoreceptors into all the light that hit them, and you create a picture, okay, that you see right now. Ciliary bodies, they are mostly ciliary muscles. These are smooth muscles, and they position the lens to focus it. That means they change the lens thickness and a little bit change the lens position in the eyeball to make sure that we go to the macula lutea of the retina. Remember, the macula lutea on the retina, macula lutea is like a target. And in the macula lutea, we're going to a bunch of cone cells here to where the point is that my vision gets the best, the closest to the center I get of the macula lutea. Ciliary muscles fold up, cause ciliary processes, and ciliary body, we mentioned in lecture, does one other thing, produces the aqueous humor. So let's take a look at these things. We saw iris. Iris is the colored part of the eye. This one here is brown. We saw the choroid, the vascular portion here, all the blood vessels you notice here. Pupil is the opening in the iris. Lens being the structure that focuses the light onto the macula lutea where the photoreceptors are. Ciliary body is the part made up of ciliary muscles inside that position and change the shape of the lens. And so we could see those. And the ciliary muscles, they help form these folds that we talked about. They help form a lot of folds called ciliary processes. And I do believe the ciliary processes folding here, you can see here's ciliary processes, here's ciliary muscles. Ciliary body has those muscles that form the processes. Okay, cool beans. Now, we also see the last tunic is the retina. Retinal tunic, retina, neural tunic, is the innermost. Now, it is got 1, 2 main layers, a pigmented epithelium and the neural portion. Now, the pigmented epithelium is going to have all the melanin in it to absorb extra light, preventing kind of reflection. Now, your dog doesn't have a pigmented epithelium. It's got a reflective layer. So when you shine light in your dog's eye, you see the dog's eyes glowing back at you. That's because it doesn't have this. It doesn't have that pigmented epithelium like we do. So they can see well at night. We can't. We are daytime creatures. We're morning. We're active in the daytime. So photoreceptors are rods and cones. That is the first component to the neural portion. Then the neural portion will have bipolar cell layer and a ganglion cell layer. Now, the neural portion, photoreceptor cell layer here, photoreceptor layer, that is your rods and cones. Now, rods are good in the dark. R for rod. R is found in dark. And it is by far, there is rowdy rods. There's a lot of them. Rowdy rods, because there's many of them, and they tell me black and white, dark, they work good in dim light. So there's rowdy rods. If you've got a rowdy group, there's a lot of people there. They're most numerous. They are found, they do black and white vision, so your shades of gray, and they work in dim light. This is why, because rods are being stimulated when there's not much light. That's why everything looks gray in the dark. Can't tell color well at all. Now, C for cone, C for color. They do color. They see color. They see red, green, and blue. So they do the colors. There are 3 types of cones. Cones do colors, red, green, and blue. So they're different wavelengths, and they are less sensitive. That means it takes more light to activate them, so they need bright light. This is why it's got to be bright for you to be able to discern different colors. Now, with red, green, blue can be put together to make all colors. These are the 3 primary colors. Red, green, blue can go on to be make into any other colors. Your primary colors can go in to make secondary, tertiary, quaternary colors of all shapes, shades, forms, etc. Now, red, green is very common to be lost, because this is actually the most common color blindness is red green color blind, especially for males. They miss the genes to cope with those proteins to do red and green. Now, cones and rods both synapse with bipolar cells. Bipolar cells synapse with your photoreceptors, rods and cones, and they also synapse with ganglion cells. Now, here's the important thing. Rods, you can have one bipolar cell synapsing with several rods. So several rods, many rods, can go to one bipolar cell. But one cone for one bipolar cell. Okay? So think of rods, since they're rowdy, they're not monogamous with bipolar cells. But your cute cones, they are monogamous. It is one cone for one bipolar cell. They're very monogamous with their bipolar cells. Ganglion cell layer, this is what goes from your bipolar cells and their axon makes the optic nerve. So let's look at that. So the retina, I'm going to zoom in here, has the pigmented layer of cells that have melanin in it. Then it has the area in it called the neural portion, neural layer, where there is a layer of bipolar cells, a layer of photoreceptors, a layer of bipolar cells, then a layer of ganglion cells. Now, remember here you can see cones are in purple, and there's not many of them, but the rowdy rods, there's lots of rods, there's a lot of them because they're rowdy, and you can have here one bipolar cell is receiving many rods, but here cones, one cone for one bipolar, one cone for one bipolar. Bipolar cell has many rods, okay? So, that's a diagram I will use on the test, or potentially could use. I could point to one of the cells. I could be what layers, what cells that. Now, the Auris Errata. What is the Auris Errata? Auris Errata, if I go and find the retina, where the vascular tunic meets the retina, forms a serrated edge of the retina where it bumps up against the vascular tunic. That's called the Auris Errata. Now, the macula lutea. I want you guys to think about the macula lutea as like a target. Now, in a target, and I kind of wish this was done here. Boop. In a target, what you have is at the center is a bullseye. Well, what happens is your macula lutea is like a target. It's going to freeze up, isn't it? I'm going to pause. Okay, it's back. There we go. So, the Auris Errata, now the macula lutea is like a target. So, at the center of the retina, cones become concentrated. As we move towards the center of the fovea centralis, this is the bullseye. So, target, bullseye, the center of the macula lutea, where there are only cones. There are no rods, only cones. This is the center of the macula lutea. Macula lutea or macula is the center, is where the light should go. Fovea centralis is where we focus that in. Then we go on, leaving the back of the retina is the optic nerve. And where the optic nerve leaves the retina forms an optic disc. And this is a blind spot because there's no rods and cones there. So if light hits the optic disc, you can't see anything. It's a blind spot. So here you can see the Auris Errata is where this thing right here, this is part of your vascular tunic, where it meets the ridge of that is Auris Errata. Macula lutea is found right here. So if we look, that's going to be the fovea centralis and the whole area around its macula lutea. There is no good image of the macula lutea except for right here's the macula or macula lutea with fovea centralis at its center. Optic nerve comes out the back, and where it does is the optic disc. Now it's best to see optic disc here and here. And lastly, we're going to talk about the cavities, and then actually we are done. And I'm going to talk a little bit about your exam, a little bit more with the senses, and then we'll do the endocrine lab, and you guys will have your first lab exam stuff done. So so here we have our cornea, here we have our lens, and these become the first landmarks to find the anterior cavity. Anterior cavity, we go behind the cornea, and in front of the lens is the anterior cavity. Then behind the lens is your posterior cavity. Now, the anterior cavity has two small chambers. The anterior chamber is behind cornea in front of the iris. As you go through the pupil, behind the iris and in front of the lens is your posterior chamber. Posterior chamber, anterior chamber. Now that is filled, your anterior cavity is filled with aqueous humor, which is made from the ciliary bodies, as we saw in lecture, and that eventually goes to the canal schlemm, sclerovenous sinus, which puts that aqueous humor back into the venous system, reducing pressure of the eyeball but that is messed up during glaucoma. Now, posterior cavity back here is full of vitreous humor. So sometimes called vitreous chamber, full of a fluid, a very thick fluid called vitreous humor. So one of the things I'm going to be doing is for the exam, the diagrams. So be ready for models and diagrams. This diagram is on the test. Neither is that. These are for practice only. But this could be on the test. Any of these images here, all could be on the test. Okay. Now go through the terms because these will be the multiple choice things I'm basing on is the terminologies where I'll have some multiple choice questions. It's usually on this one. I do believe it's 30 practical, 20 multiple choice is what I'm doing. Now, the part 2, the membranous labyrinth, vestibular complex, utricle saccule, all that good stuff. So when you're going through these things, these are terminologies you're looking at for the multiple choice questions. And then we're going to go into these blanks. And these images will be used. What I'll be giving you guys is a printout of these pictures and point to it, name this structure. Okay. So that'd be part of your examination as well. So do take turns to practice these things. Label the things that we've seen on it and practice it, making sure you know what you're doing. Now, unfortunately, the place I'm recording, I can't put the models under the document camera and show everything, but you will find under content for the Senses Lab, the ear model labeled and the eye models labeled to get the answer to everything here you need. Okay, it is these photographs and they're labeled for you that you can look at. These are the practice pieces, and I put the practice at the end of each PowerPoint for you guys to use. So this concludes the video on that, and our next one will be endocrine, and that'll be the last one for exam one for lab. And I'll talk more about how we do that. I'll have a separate video working you guys through how to prepare for that. Plus, there'll be some information in the course introduction about it. Plus, there'll be things in the schedule about it as well. So there should be a multitude of places to get information from for this course when you get there. So guys, thank you so much, and we're done.